to introduce Professor Vinck.
Thank you, Andres, for the very nice words, and thank you very much, Tristan, Professor Mullen, for organizing and inviting. It's really has been a very nice time discussing science with you guys and staying in a nice college. It's a wonderful experience. So today I will talk about a subset of the research we're doing in the lab that concerns mechanisms of attention and prediction. Okay? So if you are looking at a certain input, then you have, of course, a sort of a augmented reality in the sense that attention changes the way in which you perceive the scene, right? So you're attending to the bits that are most behaviorally relevant, and your brain, of course, also constructs certain predictions of the future, such as predicting that these two cars might crash, right? And in a very simple way, the way we think about that is that such cognitive processes result from an interaction between lower areas with higher areas, which are recurrently coupled, as well as modulatory centers that change the way in which these areas interact. And our lab tackles these sort of questions with computational and experimental approaches. In particular, we work a lot on mice, as you said, with optogenetics and high-density recordings, but also marmosets and humans. And our lab does a lot of modeling as well. So I have 4 questions today, and I think the answers to some of them might be a bit controversial and different than what you expect. And the one I will not read them all, but the thing I'm going to start with is the mechanisms of attention. And I will contrast different theories of the neuromechanism of attention, in particular firing rates and coherence. Okay? So one of a very striking observation by Desimone and colleagues, as described in this nice review by Desimone and Duncan, who also works here in Cambridge, is that stimuli in the visual cortex, they actually compete. Okay? They do not linearly summate. This means if you present 2 stimuli in the receptive field, for example, of a V4 neuron, such as here, then the response to the stimuli alone would look something like that. But we know actually if you present both stimuli, that the response is more or less the average of the 2. Okay? So they do not add, but they actually mix. And this occurs because multiple stimuli cover these large receptive fields, and that creates a competition in the ventral stream that somehow needs to be resolved. Okay? And one of the ways in which that competition might be resolved is attention. Right? So if you now have a case where attention is directed to one of those stimuli, for example, the green one, then your firing rate will approach the firing rate as if only that stimulus was presented. Right? One way to understand that is as if your receptive field is basically shrinking around the attended stimulus. So that poses the question, well, how does the cortex, when neuronal populations receive multiple inputs, and they have to selectively respond to one of them, how does it do that? Right? There have been multiple proposals about how that may work, and how attention might bias that competition. And in general, the 2 major ingredients in that equation are going to be feedforward and feedback interactions, as well as the circuit mechanisms. Because ultimately, somewhere you need to have a top-down feedback, right, that modulates activity in lower areas, and then this causes also change in the feedforward transmission, so you need to understand this from the perspective of recurrent dynamics. And in general, what's going to be important throughout this talk is that the connections between areas, as shown, for example, by Henry Kennedy or Van Essen and Velleman, is that they are organized in a laminar way. Okay? We know, of course, that the feed-forward projections from this layer 3 neurons to the higher areas, they always target the granular input layer. But feedback always arises from the extragranular layers. For example, neurons in layer 2 or neurons long-range connection in layer 6. And these connections actually exclusively go back to the supragranular and the infragranular layers. Okay? Feedback never goes to the granular layer. It skips exclusively the granular layer, which is quite interesting because there is no direct convergence of top-down and bottom-up. Okay? What's also striking is that the connections, the feed-forward and the feedback projection neurons, are actually distinct populations. So there is no overlap. Okay? The neurons that project forward to higher areas, they do not project backward. They're separate populations of neurons. And then, of course, the other thing we will consider is that the local microcircuit consists of two major component excitatory and inhibitory cells, and we will, and you have to understand the modulation of the lower circuit through the interaction between those components. And in particular, we're interested in the somatostatin cells that project to the dendrites of the neurons, so they can directly gate the excitatory synaptic inputs. And the provalbubin neurons, which basically inhibit perisomatically, and they can gate the output of the neuron sort of more globally, right? And then there are VIP neurons that regulate that circuit. And in general, feedforward transmission is usually going to the PV neurons, so the fast spiking cells. Okay, so they normalize sort of the input, whereas feedback and modulatory input tend to target more globally the other GABAergic cells. So that's a bit the background to keep in mind for some of what we're going to show today. So there have been three major proposals for neuromechanisms of attention. And the classic one is quite simple. It says that maybe, actually I did it the other way around, sir. This one should be the active one. I got it wrong. So the firing rate is higher for the attended one, and that causes somehow an increase in transmission, right? So it's somehow the salience of the stimulus that allows this stimulus to be selectively represented. So just more spikes, okay? And there's some evidence for that, although the firing rate modulation lower areas for attended stimuli are usually not that large, and they're not very consistent across studies. The second mechanism that has been proposed is that you have, rather than an increase in firing rates, you have an increase in the synchrony among the spikes, all right? In particular, at higher frequencies, such as the gamma range. There has been some early evidence for that, for example, from Fries and Desimone in 2001. But actually later studies have shown that it doesn't seem to be a consistent increase in synchrony in this gamma frequency range with attention. That leaves another mechanism, and we'll come to a fourth possible mechanism at the end of the talk, which is our proposal. But another very influential idea is that there is something happening in the communication between these brain regions. Okay? In particular, the idea is that you have, at each one of these sites, you have a certain activity fluctuations in this gamma frequency range between 30 and 80 hertz that commonly occur, and that because those fluctuations are more coherent with the receiver, you have a more effective transmission because your spikes are arriving at times where the receiver is more excitable. Okay? So this is sort of the idea of communication through coherence to explain selective communication. And there is, of course, in the visual cortex, this quite striking phenomenon that was described by Wolf Singer, who works at their institute, but also earlier Hübel and Wiesel, is that if you present visual stimuli in the visual cortex, then you see, of course, an increase in the number of spikes, but you see that the local field potential shows very characteristic rhythmic fluctuations that you can easily pick up from the local field potential at the level of memory potentials all the way up to magnetoencephalography, for example. All right? And these occur for particular stimuli that tend to be a bit larger, but they synchronize the activity across neurons in the visual cortex. And now the idea of CTC is that because you have more synchrony between the areas, you have effectively more transmission. So there has been some evidence for that. These are multi-scale recordings from V1 and V4 using ECOG recordings from Conrad Bosman and Pascal Fries, where they show in a classic attention task where the cue instructs the animal to which stimuli they have to respond, right? So a matching cue tells the monkey when it needs to respond and what it needs to ignore. So that's a classic instruction top-down attention task. You see that there is an increase in the coherence between V1 and V4, as you see here, suggesting that there is some enhanced coherence, and that has been interpreted as perhaps the mechanism to now also transmit the information selectively. Okay. So those data were all based on LFPs. So they pool a lot of neurons together, probably 10,000 or 100,000, depending what region they sample from. And actually there's basically very little data about how different cell types and different layers between areas are now interacting with each other in the context of attention, right? And we think that's critical data to have because ultimately attention is going to depend on the interactions top-down, so feed-forward and feedback between the areas, and they have a laminar organization, and you need to understand the way in which different cells are engaged. So we cooperated with Alex Thiele in this study to analyze the data from a huge number of cells recorded from V1 and V4, where you have overlapping receptive fields between the two recorded areas. And the recordings are, in this case, laminar, so you have data from each layer of the cortex that you can identify. And crucially, these isolated units, we can separate them into fast-spiking interneurons, so GABAergic neurons, and the cells that have broader waveform action potentials, namely excitatory cells, so the blue ones. Okay? And the task of the monkey here, again, at some point this cue turns on, it's a red cue, and that tells the monkey I have to now respond to the red target, which at some point is going to dim. Okay? And of course, then one of the stimuli that has this dashed circle around it is the site from which you record the neurons, right? So at least the receptor fields, the recorded cells are always in that location. So that means the stimulus can be in, the attended stimulus can be in the receptor field, or it can be outside of the receptor field. Okay? Now, what's going to happen? So here you see an example of a local field potential in V1, local field potential in V4, and you have the corresponding activity of some single units. And as you might already appreciate, you see actually more rhythmic activity in V1. And we could establish in this prep that there is a replication of several findings that have been previously made. In particular, when you have attention and you record V1 and V4 and you look at the local field potentials, so basically the local EEG, you see that there is an increase in the coherence in the gamma frequency range that is quite pronounced. Okay? So there is definitely something changing in the relation between the signals between the two areas. The other thing that you see here, when the cue comes on, the attentional cue, then you see after 150 milliseconds roundabout, you see a rise in the firing rates of the neurons in V4, and as we'll later see also in area V1. Okay? So both things are actually changing, firing rates and interareal coherence. That now poses the question, what is the relationship between these changes? Okay? And which one is actually relevant, right? Okay. Now this gamma rhythm that you have with visual stimuli, we have quite good evidence that originates from V1 and it actually propagates forward. So you're measuring that influence in V4. And it seems that in this data set, the next area, so V4 actually doesn't generate an intrinsic gamma oscillation. So on the left, it's your left, on the left figure, you see here that V1 spikes show a clear synchrony here, but actually V4 doesn't actually show intrinsic oscillatory activity. There is no peak in that phase locking spectrum. And then we also see that here the V1 neurons fire a few milliseconds before the next area, meaning there is a feed-forward propagation of this activity pattern to V4. So now comes the question, what does this gamma rhythm from V1 actually do to the cells in V4? Okay? And to our great surprise, when you look at this and you separate the neurons in the excitatory and inhibitory cells, you actually see that in the next area, so area V4, and you relate that to the signal in V1, so the LFP fluctuation V1, only the inhibitory interneurons are actually synchronized with the lower area. Okay? So there is a feed-forward signal coming in at a higher frequency that has been related to attention, but it seems to only impact the inhibitory cells, okay? Not the excitatory cells. And when you see this PPC, this is a measure of synchronization. Maybe some of you that work with EEG might know it. It's some of the work we did in our PhD thesis. Now, if you look at the anatomy, then you expect that signal from V1 to particularly affect cells that are in the granular input layer of V4, right? So layer 4, and that's indeed what we see. So the gamma signal from V1, okay, these fluctuations, they seem to drive the interneurons, the fast-spiking interneurons in the input layer of V4, but they have very little effect on the excitatory cells in area V4. So, and the signal basically then doesn't propagate beyond the input layer of V4. So the high-frequency fluctuations get to the next level, or a higher area of V4, and in that input layer they predominantly recruit inhibition, inhibitory activity. Okay. And we found that that is actually tension-specific. So when attention increases on the stimulus, the high-frequency fluctuations from area V1, okay, that drive the inhibitory cells in V4, you see that there is a stronger phase locking with attention than without attention. But you don't see any change in the excitatory cells. All right? Andres, but is there a way to turn off the screen? Can I? Or is it safe? Just my attention is sometimes going to the screen because I see myself move. I can press this, right? More peaceful. Okay, thanks. So attention affects inhibitory activity in the input layer that is phase locked to V1, but it doesn't affect the excitatory cells in V1. So there's a very specific cell type specificity of the interareal synchronization. And now when we look at the firing rates with attention, we see a very completely different pattern. So actually the firing rates, the modulation of rates that are increasing for the attended stimulus, so positive values here on the x-axis means firing rates are higher with attention than without attention. And you see that those changes are actually the strongest in the superficial layers, and they're relatively weak in the granular input layer as well as the infragranular layers. And that makes sense because in V4, feedback, top-down feedback, arrives for a large part in the superficial layers. So our idea is that the feedback directly modulates, increases the sensory responses of neurons in the superficial layers, and it skips basically the other layers. Okay. And what supports that is that we actually see that attention first acts in area V1. So the first significant firing rate enhancements are found, in V4, are first found in V4 superficial layers. As you see in this row, there's a significant effect at early latest in V4. And only later in time do you see attention effects in the lower area, so area V1, and then subsequently also in the granular input layer of V4. So it seems that attention is actually traveling backwards, as also has been reported by Bob Desimone and Bad Buffalo, backwards from higher areas to lower areas, and then because it increases the activity in the lower area, there is sort of a re-entry in the granular input layer of the higher areas. So you get a loop. Okay? Now, given that we have seen both firing rate increases and coherence changes, we could use that data to actually see which one of these mechanisms might potentially be more reliable. Okay? So which one actually allows you to discriminate the state, the attentional state of the animal from trial to trial. And it actually turns out that generally we find that firing rates are more reliable, even when we look at single neurons, than the coherence between the two areas. So the coherence between the two areas is very significant, but on a trial-by-trial basis, it actually doesn't seem to reliably discriminate attention in from attention out. Well, why is that? Well, if you think of two signals here, the same signal here is coherent, has a very low coherence, or you have a very high coherence here, you can appreciate that on a moment-to-moment basis, you need a very long integration time to actually distinguish these two cases from each other. That's because something that is incoherent will, by chance, still often have a consistent phase relationship. Okay. So, what we conclude from this are two things from this study. The first one is that there seem to be two separate routes through which attention is modulated. The first one is the enhancement of sensory responses, firing rate responses, in area V4. And that happens particularly at early latencies. It's specific to superficial layers, and it's consistent with a direct feedback modulation. And you see it in inhibitory and excitatory cells. Okay? And there is a second mechanism whereby you have an increase in the gamma rhythmic recruitment of fast spiking interneurons in the granular layer of the higher areas. So the feed-forward pathway also gets amplified, but that seems to primarily activate interneurons. Okay? It seems to be separate. It's a distinct cell type profile and a distinct laminar profile. So we think that has actually a different function, such as, for example, increasing the inhibitory tone in the receiver such that you improve the signal-to-noise ratio of your transmission. There is some evidence that actually activating interneurons in the receiving input layer can enhance your SNR by also suppressing distractor inputs. So, but in general, these data, because we see that essentially the gamma rhythm recruits inhibitory activity, it's not consistent with the CTC mechanism. Because the CTC mechanism predicts that you have fluctuation in excitability in the excitatory cells. Okay? The excitatory cells in V4 should have been phase locked to V1, and that should have accounted for the increases in firing rates, which we did not see. So there seems to be something funny with the way in which synchronization in the brain affects activity in a higher region. Right? We usually think of synchrony as something that enhances the impact of spikes on the next postsynaptic target population. But here we saw that if you have more synchrony, it seems that the effect of that is actually to activate fast spiking interneurons. So, but that contradicts this idea that generally the gain of a population is a function of either the firing rate or the synchrony. Because synchrony should lead to some increase in the membrane potential fluctuations that increase the chance to actually cross the threshold, because these EPSPs are now getting integrated better in time. Right? That's the very classic old notion from Chris Koch and Teresinyovsky. But it's not so simple, because synchrony is not something that's frequency independent. Okay? Because these fluctuations can increase at different frequency bands that might actually have different effects. So, in general, a quite influential theory, for example, Andre Bastos or Carl Friston, is that these gamma fluctuations are generally seen as a mechanism for feed-forward communication, and that they transmit perhaps the unpredicted aspects of sensory stimuli, so prediction errors, right? Which thus far has all been based on LFP signals. So you see here an example, the feed-forward, Granger causality, a measure of predicting a signal ahead between V1 and a higher area, seems to particularly peak in that gamma frequency range. Okay? But that doesn't really sit well with the conclusion that actually the gamma, that frequency, recruits primarily inhibitory activity in the higher areas. Okay? That is not consistent with the mechanism of feed-forward. And we wanted to know if that actually happens in other model systems, and if we could pin down the specific cell type of that. So what we did, for example, here in the mouse, we look at the activity from LGN that has a high-frequency rhythm and the way in which it impacts V1. And then using optogenetics, we can record from specific inhibitory cell types in area V1. Okay? And we can quantify to what extent they're phase-locked with the activity in the LGN. And what you see is that phase locking is very specific to the inhibitory neurons that sit specifically in the granular input layer, and they are pavulvimine positive. So they're a particular fast spiking interneuron basket cells that are recruited by feed-forward gamma rhythms. Okay? Another thing that we did was to look at visual flicker. You might know this story that visual flicker could perhaps lead to changes in neurodegeneration when you stimulate the brain at high frequency, as been shown by Lead Size Lab. But we actually see that when you present flicker, that there is a profound low-pass filtering, where you still have phase locking in the LGN, at 40 Hertz. You have a very little phase locking in area V1 at 40 Hertz. And in the hippocampus, there is actually no entrainment to these high-frequency flicker stimuli. Actually, the lab of Yuri Bijaki recently showed the same thing, and they actually showed also lack of effects of neurodegeneration on CA1. And we could also show in this study that, again, this is specific, whatever you see in V1, to the fast-spiking interneurons. Now, finally, we did a study where we recorded from two areas simultaneous, V1 and V2, and we optogenetically stimulated the lower area, V1, at different frequencies. And we wanted to know which frequencies in the brain might actually propagate. So these are connected, they have overlapping receptive fields, and as you see here, the low frequencies propagate to the higher areas, but when you synchronize the lower area at a high frequency, it doesn't propagate to the next area. Okay? So in general, synchrony at high frequencies basically primarily recruits inhibitory activity in downstream targets. Low frequencies tend to propagate across multiple levels, and that contradicts the idea of gamma mediating prediction errors, okay? Because the primary effect appears to be inhibitory. Now, what explains that biophysically? Well, we know, let's say, if you have presynaptic population in V1 that is gamma synchronized. When these inputs arrive in a postsynaptic target population, there is, of course, a profound low-pass filtering that's due to the capacitive membrane properties of the neurons. Okay? So these fluctuations at the level of the VM are going to dampen heavily. And we know that the transfer functions of excitatory cells, they are much steeper than of interneurons. Interneurons are able to follow more high-frequency inputs. Excitatory cells are primarily driven by low-frequency inputs. They often have a resonance in the theta frequency range. That means that if you look at the VM of the neurons in the next area, those fluctuations here are going to be massively dampened, okay? Because they're filtered. And much more so in the excitatory cell, that dampening, than in the inhibitory cell. So that leads to some phase locking in the GABAergic cells, but you now do not have propagation across levels because the inhibitory cells do not project to the next layer, etc. Okay? So it basically stops sort of the communication. Now, that leads to a surprising idea. Okay, well, if the gamma is not mediating prediction errors, then, as people have said, then what is it actually doing, right? So I want to talk about that in the next slides a little bit. And actually, when Friston and Bastos put forward that idea of gamma and prediction errors, we published a review paper in 2016 where we actually speculated that exactly the opposite might be true, namely that gamma occurs when stimuli are actually predicted, both temporally and spatially. There was a study with Conrado Bosman. And we recently did two studies where we tested that idea, and we presented stimuli to the monkey, so natural images or colored surfaces, and we recorded the activity in area V1. And we see actually that the more spatial predictability there is, meaning you have a stimulus that can be better predicted from the surround, okay? More spatial predictability, the stronger you see these gamma rhythmic fluctuations in the early visual cortex. Okay? As you see here from the spectrum, more predictability, and then you see the emergence of a gamma rhythm. Okay? And here is an example. If you have a homogeneous surface where every colored input is exactly the same at every location, this is a 6-degree stimulus, so fairly large. You see a very nice rhythm, but when you present a deviant stimulus at the site where you record from the neurons, you see that you completely disrupt the rhythm, okay? Now, that means that we found we found a gamma rhythm for stimuli that have a very high redundancy, that are very much compressible, and that typically actually have a lower image salience, okay? Locally, there is less salience. When you look at this stimulus here, it's much more salient, that location, but you have an absence of a rhythm. Here, there is much less salience because the information is the same as everywhere else. And that actually kind of makes sense. If something is very redundant, you maybe want to actually recruit inhibitory activity in the next brain area. Okay? The same happens with temporal predictability. So when you now look at the activity in the visual cortex, and you present, for example, predictable motion here, and you compare that to a bar that is moving in an unpredictable manner, then you see here that you get a gamma rhythm, but here the cells basically follow the structure of the external stimulus, which is unpredictable, but you see absolutely no gamma rhythm here. Now, in general, you may appreciate that the onset of such rhythms typically happens way after the first feed-forward transient, right? So if you present a new image, the first 100 milliseconds, the activity is dominated by a more broadband aperiodic signal than an ERP, you know. And then after that, you see that the activity of the cells gets more sparse, so the cells' defining rate activity decreases, and you see the onset of such rhythms. And that led us to hypothesize that what's perhaps happening is that the cortex generally operates in two sort of distinct regimes. One where you have, let's say, an unexpected input or an unpredicted input, and that input leads to a transient that propagates across many levels. Okay? So you have here an amplification through some aperiodic signal that gets amplified through some nonlinear dynamics. But at some point, when the brain, let's say, makes sense of the input, so when it becomes predictable and the inference process stops, this is when you typically see the emergence of these rhythms at a later onset. Okay? And typically there, we have also quantified that, the firing rates tend to be much more stationary. Okay? So here you have sort of an equilibrium state, and in these equilibrium states, it's where you tend to see those rhythms. Now, that could actually make sense from the perspective of predictive processing because in this late phase, your bottom-up inputs actually are matched by top-down predictions, all right? And that actually leads to a tighter balance between excitation and inhibition in the local circuit. And so that tight balance typically is actually associated with, for example, the emergence of gamma and beta rhythms. And models also suggest that in that regime, these rhythms may facilitate the sustained and efficient encoding of information in the local circuit. For example, work by Matthew Chalk and Sophie Deneuve. So bottom line, we do not think of these rhythms as conduits for feed-forward signaling, but rather as a sustained signal during equilibrium states. Okay? Now, that comes to the last part of my talk, namely, what's the relationship between structural and functional connectivity? I think that's something that probably each one of you, even if you work with fMRI, is interested in, because you have all kinds of networks and you want to relate them to the anatomy, right? And there seems to be some major discrepancies here in what I've just told you, right? So on the one hand, we said that measurements of local field potentials, which probably also correlate well with Bolt signals, they seem to suggest that these higher frequency rhythms are going in the feed-forward direction. Right? At the same time, we said, well, it's maybe actually the opposite, because the higher frequency rhythms recruit inhibitory activity in the next areas. It's the lower frequencies that actually do seem to propagate. And the empirical data generally doesn't seem to be consistent with this communication through coherence framework. Okay? So that led us actually to think that there might be an opposite causal relationship here between the variables. So the classic idea is that you have some top-down feedback, let's say with attention, that modulates the coherence between brain areas, right? So you have sort of a flexible synchronization, as also Varela describes it, and that modulates the communication between areas. So you get an effective connectivity because of these coherent relationships, and that's sort of superimposed onto the anatomical substrate. But there might be a more simple possibility, namely, you are changing the communication between brain areas, and that actually leads to a change in the coherence, because coherence measures that communication. Okay? So we call that basically coherence through the communication. And we developed a mathematical model to predict that. But I'm just going to show you an example of how LFP signals can actually diverge from spiking signals. These are dual recordings from arrays in area 7B, parietal region, and premotor region F5. That's from the lab of Hans Scherberger. And when you look at these both areas, you see a very strong beta rhythm in parietal cortex, and that's much weaker in area F5, premotor. And when you look at the spikes, you see in area 7B, parietal, you see a clear phase locking peak of the spikes in area parietal cortex, but you do not see that in area F5. Okay? And when you now look at the inter-area coherence between LFPs, there seems to be a clear, very clear peak in that spectrum around that beta frequency. So when you look at this LFP, you would say, there is a coupling of oscillators in the two areas. But when you analyze the spikes in area F5, then it turns out that they're actually not phase locked to area 7B. Meaning, if you think back what the LFP is, right? The LFP reflects ultimately transmembrane currents that are generated that trace back to synaptic currents, so iPSCs, EPSCs, okay? And these can be subthreshold, right? So the return currents give rise to the LFP, and those inhibitory excitatory currents coming from area 7B, they can remain subthreshold, and they might not actually entrain the spikes in that receiver. Now, that actually generally makes sense because every brain area, the spikes of each brain area, give rise locally via recurrent connections through the local field potential. Okay? But they also project to the other area with some coupling weight. And in that receiver, the measured field potential is going to be a mixture of its intrinsic activity here, eta, plus the input from the first area times some connection weight. Okay? And that's the measured signal in that area. That means that this area here contains some linear copy of the activity of the sender, right? Because of the communication. Now keep in mind that coherence is a measure of explained variance, linear explained variance per frequency. So in these situations, because of the communication, you already expect to see a coherence that emerges from that. And we performed the mathematics on this. This is the more simple model version, but we could generally show that coherence scales with the strength of the connection weight between areas and the amount of power that the sending area has at a certain frequency relative to the receiver. Okay? So just to give you an example, we have here a sending area 1, and that sending area 1 shows some oscillation at 20 hertz. And the receiver does not, so the receiver doesn't have any intrinsic oscillation. And now the signal gets projected with some coupling weight. And you see here that there is a coherence peak or Granger causality peak, even though you have a flat transfer function. Okay? And in the receiver, you do not necessarily see any signature of that input, but you do measure a clear coherence. So this is coherence without essentially having intrinsic oscillations that reflects the anatomical connectivity. Okay? And that reminds very much of the earlier results I've shown between V1 and V4, where you had this input gamma rhythm to a receiver that did not have this intrinsic rhythm. Okay? Now that model, we have applied that to several model systems. I'm just going to show you one example. You have here coherence changes between task phases of a working memory task in the monkey between area 7B and F5. So you see during fixation or memory or movement, you have quite strong changes in coherence. But you can see that they actually correlate quite well to the fluctuations of power in area 7B. And if you fit the model to this data, then you actually see that the connection weight is basically the same between the three periods. Okay? So the effective communication doesn't seem to change, even though the functional one is changing, and that's because of the amount of power in the sending region that you see here. And we could show that actually both monkeys here have very similar connection weights despite having very different levels of coherence. Now we've used that to predict, at least we published recently a paper where we showed that the anatomical connectivity in the brain, so the connection strength that's based on retrograde tracers, correlates quite strongly to the functional connectivity between the brain regions. So the more they are connected, the more you also see coherence specifically in the gamma frequency range. which matches very well with our model because it's proportional. The functional connectivity is proportional to the connection strength between the regions. To directly test this model, we actually wanted to show that if we have two areas, a sender, for example here LGN, and a receiver, for example V1, that we can get coherence between two brain areas, measuring basically the LFPs in V1 that reflects inputs, postsynaptic potentials, even when we suppress the activity in V1. Okay? So we used here an activation of specific interneurons that silence the activity of the excitatory cells and inhibitory cells in V1, not in LGN, just in V1. And then we make dual recordings with neuropixels from LGN and area V1. And we see that in the control condition, you have a coherence that peaks in the granular layer where the LGN input signal arrives. But then if you suppress the spiking of V1 cells, you still see that coherence. And that's because the LGN inputs are still arriving into V1 that are generating that coherence. Okay? So it is possible to get coherence without any effect on the postsynaptic neurons. And we could also use that model to make a prediction about gamma feedforward and beta feedback, because it actually turns out that the regions that show feedforward connections to higher areas are the regions that tend to have a very strong elevation of power in the frequency band. So for example, early visual cortex has strong gamma oscillations, whereas the areas that show feedback at lower frequencies, for example, beta and alpha, that are at intermediate levels of the hierarchy around parietal cortex, they tend to have a beta frequency power. And in such a situation, our model predicts that asymmetry quite nicely. Okay? So I have some time to go back to the question what the mechanism of attention is. Okay? So we said in the beginning that modulating the activity in the lower area, just increasing the intensity, doesn't seem to be really supported by the evidence, because typically firing rate changes in lower area tend to be relatively weak or inconsistent. There's also no clear evidence that attention modulates the synchrony in the lower areas and thereby enhances the impact. And we've given several arguments why basically the change in coherence between the areas might actually be more of a consequence rather than a mechanism for attention, especially due to the association with the recruitment of inhibitory activity. So, in general, something to keep in mind is when you look at things like coherence, it captures mostly linear relationships between signals. Okay? That means you're only capturing a scaling or a phase shifting of a signal at the same frequency. There are no interactions between frequencies. Whereas in the brain, of course, many of the interactions that we have are actually nonlinear. Right? That means you're transferring energy between frequencies, because in the end, ultimately, any form of pattern recognition, extraction, whether it's completion or separation, depends on nonlinear transformations. That's a very basic fundamental of how neural networks work. Right? And in this review paper, actually with Andres that we wrote, we proposed a model of how attention might actually be resolved through nonlinear recurrent dynamics at the level of a receiver. So in this case, we have two inputs, so the classic bias competition model, and these two inputs are mixed in this receiving recurrent neural network. And we imagine that there is some top-down module, and that top-down module provides some weak feedback to the receiving area, and it tells you which one of these two stimuli needs to be attended. Okay? And the hypothesis is that that feedback will change basically the recurrent dynamics in that RNN and change the attractor state, essentially, of that recurrent neural network. So we recently modeled that. We trained spiking recurrent neural networks to classify spoken digits. Okay? And in this case, there are two sending areas. And the task of the network is to classify the digit that is attended. Okay? So one of the two, if it's a 5 or a 6, that will be the correct output of the network. So it has to ignore the other digit. And the outputs of these two networks converge into one receiver. And that receiver has conflict. It has competing inputs. But it also receives a top-down input from a higher area that tells it which location, which one of the two inputs should be attended. Okay, the top-down module doesn't know, of course, what is the digit in this case. And then we have a linear readout of this network to do the classification. And we train the entire network with all the RNN modules that consists of linear integrated fire neurons through backpropagation through time. And now the question is, can such a network actually resolve the competition? And the answer is actually yes. So when attention biases, when basically the top-down feedback is enough to get into the receiver recurrent neural network and change essentially the dynamics of the population to an entirely different state of the state space. Okay, so here this would be attend to one stimulus and this would be attend to another stimulus, and these are population vectors in the PCA space. So the top-down input can be modulatory, and it changes the trajectory of that population and causes it to switch. And what we see now is because you have an effectively stronger integration of one input stream, we also measure an increase in the coherence as a consequence of this effective information transmission. Okay, so again, the same principle. The change in the coherence reflects a change in the effective communication between the areas. So this is in a way, we think this is a much simpler mechanism because it allows attention to operate at the site where you experience the conflict. It doesn't have to modulate all of the neurons in the lower areas, but it solves competition essentially at the higher areas where you have multiple converging inputs. And this is also consistent with our empirical observations because we see that the attention effects are stronger in the layer where you have basically top-down feedback, so in the superficial layers, right? And it also predicts the increase that you might have in the coherence ultimately, as we show here. So with that, I think I'm on time. Yeah, and I would like to thank, of course, the lab, and particularly Jorgis Peropoulos. He's not in the picture, but he did much of this attention study. Marius Schneider, he developed his communication, coherence to communication framework, and also the Flickr study. And then Atani Etsanu, our lab manager, and then some of the collaborators, including Alex Thiele-Hanscheber from DPZ, at the AZ Wolf Zinger, who has been great support and mentor, and then, of course, the funders that funded this work. So I'd like to thank you for being here.
Whoever needs to leave. Whoever is brave enough to stay for the discussion. Timothy.
I'm going to call Paula because she's really slow in the car to pick up Nico.
It's a car.
Yeah, she's coming back to pick up Nico, but if she's coming late,
Ah.
rush.
Okay. Okay.
Which is not what I want.
Questions, please. Any questions?
Yeah. You showed path of the effect of attention that went from top to the bottom. And I wonder, how do you think this, where do you think this attention path starts? Like if you trace it, where would it?
Right. There's evidence that, for example, frontal eye fields from Tyrion Moore plays an important role in that. So that if you do FEF stimulation or also prefrontal lesions, that you really affect the attention in the lower area. So that would be the main candidate for such projections. Right? And the frontal eye fields, of course, has very strong attentional modulations, right? So that's a likely candidate.
And what would modulate them?
They identify, of course, it's top-down attention. So there's some cue that instructs the animal, so that those neurons must somehow figure out in the frontal cortex the rule, and then have to map onto a specific output signal that goes to the lower area, right? But, it actually doesn't have to, as long as this signal is specific, if it changes the activity pattern in the higher area, depending what you should attend, it will actually work. So, you don't need a very specialized mechanism to solve attention, right? You can do it with standard recurrent networks. Thanks.
There's one there.
Oh, yeah. I was wondering about the kind of division between the equilibrium state and the earlier, actual integration of, let's say, prediction errors.
Mm-hmm.
So in the case of prediction errors, you should see some kind of nonlinear integration or something going on, and followed by this equilibrium state. That's kind of the result.
Right.
Prediction state.
Yeah.
So it would be kind of interesting to show if you could somehow that you have this kind of integration or nonlinear stuff that happens before you get the like steady oscillations
Mhm.
in the corresponding different time points. Is there anything done on that?
Not
Seems like
not really. I mean, if you it
kind of support the idea.
right we are working on that. So you would have to analyze multiple areas and then actually indeed show that you get a rapid ping pong between these areas, and at some point you get sort of a convergence to a particular inference, right? And that here you get sort of these rhythmic states, and that should lead to profound difference in connectivity and also linear and nonlinear measures. But yeah, that we will have to do.
Yeah, no, but it seems like based on that you should always get like some kind of phase one, which is the prediction error.
Right.
Resolving itself followed by this equilibrium.
Exactly. That's the idea, that actually the prediction errors are sort of signal to transients. Anders' work also speaks to that, and that these, that propagate, at least we think, through really nonlinear dynamics in recurrent networks, and then it's and at some point, if your fine rates get more stationary, then you will have an equilibrium, right? And this is where you typically see the gamma or beta rhythms. And in that stage, you might have more cognitive signals that come in to perhaps modulate the expectancy to the next stimulus or something like this, right? Yeah.
So, are you familiar with some of these studies that have entrained oscillations with, for example, TMS, and show
Right.
Perceptual consequences and behavioural consequences,
Mm-hmm.
and how that fits into your...
Right. Yeah. So there are. These studies have typically, I think, the TMS ones stimulate at alpha or something, or beta, right? So as far as I know, the frequency where you get sort of the perceptual effects tends to be at a more low frequency. There's also, for example, work from John Reynolds showing that if you optogenetically stimulate V1 and you have the monkeys doing a task, that you get a distortion of performance in that task if you stimulate at low frequencies. But if you stimulate at higher frequencies, you do not seem to affect behavior. I think also there is some work showing that more at low frequencies, at least in the visual system, you tend to have hallucinations or psychophysical illusions, and that they do not happen at the higher frequencies. As far as I know, you probably know this better than I do. But so that's and then of course there's some work from more. There's some interesting work from screams. That auditory signals, that screams tend to have a lot of energy in this higher frequency range. So they are kind of aversive. And we also see, this seems to be also a principle in the visual cortex, that stimuli that have these generate this high frequency activity tend to have some aversive quality to them, like grating stimuli are kind of cause visual discomfort, right? And well, the other thing is that there are some external stimuli that, for example, auditory cortex and trains to, but that typically also happens more at lower frequencies, like theta or something, right? So, yeah. But I do not know this TMS literature that well, I must confess. So.
Neither do I.
Yeah.
I have
Yeah?
I have seen right, but I'll ask why. So when I started the PhD, it was very like, okay, let's read this Vinck et al. 2011 that shows that phase synchronization can be measured with this WPLI metric. So basically, and I have papers showing that there are at least good evidence for thinking that in certain cases, like when you're imagining stimuli or whatever, there is some kind of effects on these linear metrics at the EEG level. So what I've been wondering since we met last year is what the hell are we actually measuring in the EEG when we compute coherence of WPLI, and there are effects in the gamma range. Like, can we actually translate these results of, okay, let's think of coherence or any linear metric as just a consequence of communication? Is it that immediately translatable to what happens at the EEG or the MEG level? So what's your take on how generalizable is the Schneider et al. Effect to EEG or MEG?
I think that depends a lot on the specific model, but I mean, if your coherence values are relatively weak and you have these sort of power asymmetries where you have a certain source of a power and you then look at its consequences, when we look at the data, for example, from Charlie Gray, who has these recordings of LFPs, okay, they're intracranial across the whole brain, you see that for many connections our model matches very well with what you are seeing. But there could be, of course, some exceptions when you have, actually really strong oscillators in multiple sites that could have some coupling that is genuine synchronization, right? But then typically you will see a very, very high level of synchrony. For example, subfields of the hippocampus, when you look at the rodent theta or something, might follow in that kind of a model. I see here some power issue. But in general, the model, it's Occam's razor would say that this is a simple model. So I would think it's a kind of a safe assumption that.
The curse of the UK.
Yeah.
Turn on.
Right. At least we have tested the model in many cases. Also the V1, V4 case. We have, I didn't show the study here, but we can actually model that data very well from changes that happen just in the sending region. So it is actually, I think it's quite possible to infer structural connectivity from functional one, which is quite useful in humans. But it's, I think, very difficult to infer effective communication from the model, because you have to control for all of the other factors like power, frequency, what have you. So it's very hard to interpret functional connectivity.
So I guess what I was trying to ask is whether people should start forgetting about the idea that when they compute coherence or PLI or brain causality in EEG or MEG, they're actually computing integration.
I would agree with that. I think it's more simple and safe to assume that a lot of this, what you're observing, is the effect of linear signal transmission. Right? So that it's probably much more interesting to look at nonlinear measures, because they are actually will reflect, by definition, some form of integration. Right? Anything that's nonlinear will measure a relationship across frequencies, and these nonlinearities, they must reflect some form of integration that at least tells you something about the signal affecting the outputs in a specific way, right? Whereas a linear signal transmission can also just be sub-threshold, right? But yeah.
I'll ask one of the 5. Maybe there was someone.
You want to have drinks, I think.
I was thinking, with the model, how would you think about the expected event that didn't happen? You know, the discussion of the omission that it comes again and again.
Mm-hmm.
We always trying to say, okay, there's a prediction, an expectation, attention, is applying a rule. But sometimes we have a few experiments, in the literature, quite a few where, the monkey, the animal, the human expects something to happen and it doesn't happen.
Mm-hmm.
So, is that can be resolved at the, as you were, answering Sharon, at the higher level, and there will be in principle no involvement, or there's no need for an integration at the lower level because there was no input. It was just the violation of the expectation of the input.
Mm-hmm. That's actually not so. Don't think that's known. There are some studies in mice from Massimo Scanziani showing that if you have a stimulus, that let's say you have a big grating, but you're blocking out the center of the stimulus that is in the receptive field. So the neurons do not have what they see in, let's say, just a screen, a gray background screen, and you only present the surround, like just the annulus. Then at least in the mouse, you see that even though you don't have an input, you have, let's say, 20 milliseconds later than normal, the feed-forward pathway, you get an increase in the activity at that site. And that's known to depend on the feedback. Right? So we know it's feedback dependent from Scanziani's work. But so that's kind of an emission stimulus, right? But of course the emission is embedded in the context that identifies it as an emission. That depends on feedback, but it's not known whether that actually the comparator circuit sits in the lower area or the higher one. So that it's testable to do that, but I think in general, I think the idea that necessarily predictions have to flow down is not really. I think it's an open question whether prediction or error signals are flowing down. And we have actually some data suggesting that firing rates in V1, if you look at the way in which they encode prediction errors, the firing rates, they do not follow low-level prediction errors. They follow actually violations of the locus of the structure in the image of high-level features. And those high-level features correlate best to deep layers of a neural network. We showed this in the Iran study. So it seems to suggest that V1 neurons are modulated by something that they cannot compute because they encode prediction error signals about something, about features that they do not represent themselves. They are semantic features, so high-level features. Okay, and then of course salience in general is something much more high-level, right? So I think this suggests the comparator circuit might actually not sit in V1. It could be that the error actually flows down. And another study that suggested this from Kasper Schwiedrich and Vinck Freiwald. They have recorded from 3 levels of the facial areas in the temporal lobe. And they see that in the lower facial area, the neurons are not identity selective. They don't encode the feature, but they encode the orientation of the face, okay? So whether it's like that or that. But they are modulated by the predictability of the identity, which is encoded in a higher area. So it seems that something in a higher level sees an error, sees there is a surprise, and then modulates the lower area to amplify that surprising signal. So I think the Friston kind of circuit, or Raoult-Ballard, where you always have the comparator integrating the bottom up and the top down, it might be quite different. It might be that the error signals actually flow downwards.
So in your final model, that would in principle be resolved between the higher level and the middle level, maybe flowing something to the early level, but not necessary.
Right. Yeah. Exactly. So the lower level, that we see also that ultimately, for example, with attention, V4 gets modulated and V1 gets modulated later on, right? And that signal seems to be a consequence of what's happening in the higher level, right? That causes some change again in feed-forward communication.
But your assumption would be that even if you optogenetically kill what's coming to V1, that would make no difference for behavior, right? It's almost like a echo.
Not in the late phase, right? So if you initially, when the stimulus is presented, block inputs to V1, then the higher areas won't have the information. But later on, it should not really, And that, again, there is some evidence from Scanziani that suggests it, because if you optogenetically inhibit the first spikes from V1, you get a big behavioral effect on discrimination. But if you inhibit the later spikes in V1, it doesn't seem to really matter, right? So, and that's probably because the circuit is just in some equilibrium with sparse coding, and there are no updates. It's just online. It's sort of show a lot of rhythmic activity. That's at least my view. So.
Comments, questions? You can throw bottles, or we have bottles in the pub. Yeah. If you've been hesitant of asking a question, come and ask a question in the pub, right? Directly with a beer. Good.
Thank you.
Thank you.